Yeah, today I'm going to have a talk about real-time monitoring of dogs and horses. And we also will have a live demo here with measuring EEG from our subject Remy, which is walking around behind me during my presentation right now. And in the end, we will have this live demo. So I will start with a short introduction about animal EEG in general, with a focus on dog and horse EEG. I will explain our device that we will be using for our measurements, the BCI core, and then I will follow with the dog EEG experiments that we did and the horse EEG experiments with some results. And in the end, you will see a live demo with EEG measurement on Remy. So to start with basic introduction, the state of the art in canine in dog EEG looks like as in this slide, as in the pictures. So typically it's for EEG cable systems, stationary systems. The animal partly needs to be sedated, and yeah, it's like the usability in the field, let's say, is not really given with systems like that. Also comparable, the MRI, similar to humans, the setup is very limiting for the animals. And shortly for the anatomy of the dog's brain, we select our EEG positions accordingly. Of course, we have here different to the human, so we need to select our EEG electrode positions accordingly. For the devices that we use, we selected here the 8 positions that you can see in the left figure. They are standard positions already marked from literature, and we try to follow this guideline with this positioning. And you see also on the right side here the skull with the brain from the MRI of a Doberman. And according to that, of course, we try to capture as much brain activation as close as we can on the scalp. When it comes to equine, so horse EEG, we have similar studies that are being done. So often sedated, semi-invasive to measure the EEG, also with computed tomography additionally. And what we try to do is to make it in a real world setting with free movement. Similarly, for the horse anatomy, we had to select standard position that were already researched in literature. So there is a proper amount of literature about horse EEG, and we selected here these 8 positions marked in red for our electrodes. Interestingly, also maybe to see here on the figure on the right side, you can see the orbital cavity and the cranial cavity, and how much distance there is in between, and that actually in between the eyes here there is no brain, so we need to really go upwards towards the ears to get here our EEG. And the device that we use for the measurements is the unicorn BCI Core-8, as you can see in this slide, and you have maybe already seen in other presentations at the spring school. Actually, we did already a measurement with Santi at the hyperscanning today in the morning. So maybe you've seen it. For humans, the device looks as so. So we have this very small wireless device that can be used with standard cap and our hybrid electrodes. The device offers a base station and the BCI core, the amplifier, that is just plugged on top with magnets, and with that you can start recording. And the same device we also use for our animal EEG measurement. Shortly, the specs for the device: it's wireless Bluetooth low energy device. It offers 8 channels, 24-bit resolution, and 110 input range. And we have a sampling rate of 250 hertz. And you can record with over 6 hours with this very small device. I also have the size of the amplifier itself, and the charging time of the device is roughly. As I already mentioned, it's very light and has a small form factor, and we offer different electrodes with the device, so it's adaptable for usage. So the ones that we use today, I will also show you in the slide, are gold cup electrodes that you may know. Clinical EEG recordings But also in animal EEG, it's often preferred to use. And this is the base station with our 8-channel gold cup. Yes, and as I said, it can be used for humans. In animals. What we did until now is measure dogs and horses, but we're looking forward to test the system on a lot of different species and to see results there. To give you an overview how the system looks on a dog, here is a picture of our company with the hybrid electrodes. So hybrid in this case means the electrodes can be used dry and with gel. Here with dog EEG, we prefer them to use gel, and the base station and the amplifier are just mounted here on the dog's collar. So quite simple setup, and with that you can measure here 4 channels EEG on the dog's scalp, and also you can use the gold cup electrodes that I mentioned before. So if you use a base station that offers gold cup electrodes, you will use conductive gel, for example, this LFX gel, to mount the electrodes on the scalp. So basically the procedure is you remove the hair and you stick the electrodes below as much as you can on the scalp directly, and then put the hair back on top. And with that, you have actually a very nice fixation of the electrodes. You can use additionally tapes or other caps to fix the electrodes, but actually by placing them with this very sticky gel, they are placed very clear. So we have this for the dogs, and for the horses, we have a similar setup. And in this case, we have an 8-channel device with, again, hybrid electrodes. I will show you also here on my table. So we have, it's just a very simple cap with a strap that you mount on top of the scalp of the horse. And we have this base station centered here in the middle, and the hybrid electrodes that can be filled with standard EEG gel and that deliver our EEG. On top of that comes the amplifier. That's how it works. So you just mount it on there with magnets. And another option, and this will also be the option that we will test today, we have the gold cups for horses as well. So we have here a similar procedure. You put the active gel, you remove the hair, and then place them nicely. You can put the hair back on top, so they're prepared nicely and ready for measurement. As a software environment for our animal EEG, we offer a similar environment as for the human EEG with the BCI Core-8. So you can use it in the g.tec suite with the BCI recorder, BCI recorder, HiSys, and GBS Analyze on Windows, and also with the G-pipe that you have seen already throughout the spring school. Here a short example of paradigm that I will also that we did already with the animals. So we did it with horses and dogs, an auditory EP oddball paradigm. In humans as a comparison, as you can see here on this slide, we have a behavior. So we have like target and non-target auditory stimuli, so beeps in different frequencies. And you have, for example, a ratio of 5:1 of target and non-target. So 5 non-target stimuli and 1 target stimuli. And if you then like play this auditory file, you average the stimuli, you get this auditory potential response here in humans. On FZ, it's, for example, 30, 50, and 100, and 100 here in this case, in a range with 0.1 seconds here as scaling and 3 microns, typically for humans. And very similarly, that's what we found also with our tests with a similar paradigm in dogs. So we again had an oddball auditory paradigm with target and non-target stimuli. And here, if you average the target stimuli, We have quite some high amplitude. This is also due to a lower amount of target stimuli, but a very similar timely behavior, and with even a stronger response as we found. And here you can see an overview of the topography of these EPs on the scalp plot of the dog in this case. So what we do is we place our 8 electrodes on the standard positions as mentioned in literature, and we place the reference and ground, which is the shared electrode behind the ear, and we potentials, and you can see here the responses with the target responses in blue and the non-target in orange, averaged across 50 target and around 250 non-target. You can see here these amplitudes. Very strong amplitude here on the right lateral side and diminishing towards the back, which we found in most of the dogs that we measured actually. Again, here the device that we used, so the BCI Core-8 with the gold cup base station. The electrodes you can see in detail here on, and in part D, you see how we attach that. We analyzed the power spectrum, so we wanted to see here comparison to EEG in humans, for example. Like where are we here with the power spectrum? Very typical behavior declines towards higher frequencies. We have here a 50 Hertz notch filter, and we also used some ICA to remove movement artifacts, because especially in dogs, of course, you have a lot of movement if you have them free moving, and that's what we try to measure. So we put on the system, play the paradigm, but they're still free to move, and this is what we also need to acknowledge in our data analysis to remove movement artifacts in the EEG. So we have here a bit higher frequency components in the raw spectrum, and when we use the ICA by removing the movement artifact, this lowers a bit here for all 8 channels in one dog. And these are the results for EP measurements in 4 dogs. So we had different dogs here participating in this experiment, and of course also we had a different amount of target and non-target that we could use for analysis, because there has to be some removing of certain target and non-target stimuli because of movement. So what we have here, you have it mentioned in each panel, the targets, non-targets that we use plotting, and you can clearly see the behavior that I mentioned before in dog 1 and dog 4, particularly also in channel 6 here, the highest amplitude. Always the plot with the channel with the highest amplitude that we use plotting. Also dog 2 shows here a clear N120, similar to the other dogs. Also similarly, dog 3 shows a behavior like that, but we had just less trials in this case, and also more. So this is something that we analyzed from 4 dogs, and quite promising to proceed with that. And now I want to show you some EEG from the horse that we did some test measurements. So we have here an 8-channel EEG trace with the hybrid electrodes, and we use this to analyze in the end events. So with GBS Analyze, for example, you can here do a video sync. So this is what we also did in this case. So you can record a video, and that's also what a lot of researchers want to do. They record the animals, for example, in their standard sleeping behavior. They often measure ECG, of course, also additionally, and other parameters. And then they measure here, for example, the EEG in the sleeping environment. And it's very important that you have here a video for synchronizing that gives you all the important markers for your analysis. In general, very important with animals, because the behavior is not as controlled as with humans, of course. So asking them to sit at the table and perform the experiment or the task, or also to lie in the bed and do the measurement, it's not working same way. And similarly with an auditory evoked potential experiment with target and non-target, and you can see here the evoked potentials from 2 channels. We have also quite high amplitudes, particularly, and we see here a positive peak at around 100 milliseconds. And maybe in the background you hear Creamy grassing. She is already excited for the experimenting. So this is what we found with our auditory nut. And in general, the recording software that we use looks like that. So we have the BCI core recorder. This is also the one that I'm going to use for recording the EEG today. We have 8 channels, for example, in this scope. And the blue vertical lines that you see are here the triggers from an experiment. So in this case, for example, we have auditory experiment. So with each auditory beep, we get a trigger and it's aligned with the data also. And it's then saved offline and also can be used online, for example, by streaming the data in HISIS and then perform processing online here in g.tec. So this is the standard software procedure that we use. And also with UDP and LSL, you can stream the data on other platforms, of course, which is also often used to further process. And one interesting part when it comes to movement, also in animals, particularly the artifact you Creamy, is the OSCAR software, so the artifact removal that we provide. So particularly interesting also work with animals. So this is also something that we are particularly working on, right? And with that, I actually finished already my presentation part. And now I will stop sharing the screen, and I asked Sarah to help me. She will hold the camera and film the horse. Meanwhile, I will set up the device. As I showed you shortly, I'm setting up this 8-channel Gold Cup device. So I'm going to mount it here on the leather strap on the horse, and then stick with this LFX gel electrodes to scalp, and then we'll record some EEG, and I showed you how this. And now stopping to share the screen, and I hope you can see the camera that starts to take. So go around and film what I'm doing here in general. So you can see here Dreamy, she's already here a while. She was grassing a lot. I guess she's very happy because she had a lot of nice fresh grass here from the garden at g.tec. And we already did a couple of measurements with Dreamy, so she knows what's going on. She's a bit hungry. So I'll put her the base station here on the back, and then I'll stick the electrodes. For that, I also need to remove a bit the hair. But let's first try to put the base station on here, the leather strap, which I need to lift up a bit. So with this base station mounting, so we use a very simple Velcro strap. With this mounting strap, I actually have the option to mount it on a lot of different positions, which is particularly important also because depending on your measurement, you want to place it on different positions. So it's not just enough to build one cap with fixed positions. So you may need here another setup. So that's how we mount the base station. You can see here the electrodes. So what I'm doing now, I'm just removing the hair a bit behind the ear, and I'm starting to gel. So here on the right side, on the left side, we have 1 to 4. And on the right side, we have 5 to 8 and the ground. So what I'm doing is, as I mentioned before, I'll remove a bit the hair. I'm sticking it on, and without even using any other tape, I can just leave it like that. Of course, in the end, I can fix and mount a bit the electrodes with, or the cables, particularly with some tape, to have a more fixed setup. But like so, it's actually super easy and works pretty nice. So I try to keep the positions that I started to press the video, but how is it still here? Yes. So I try to keep the position, the ones that I showed you before, because this is quite important also that we can make the stand up. Right. So now we are back. We have 3 electrodes positioned. Now we have number. And what I'm going to do, better, what I'm going to do is just tape cables a bit here to the strap so they don't move so much. So we remove a bit movement artifact, and with that it's a bit easier. Okay, so we now go to the other side and do the same with the other. Stream is very nice, and it's very easy with today. Until so I'll try to place them now a bit more centrally. I was a bit far, I have to say, here with this row, but now I'm going to be fully and 6. Now you can see here I try to remove here the hair a bit. Also, of course, also Dreamy doesn't have too much hair, so it's quite easy. And with dogs, sometimes you really need to lift up the hair. We have number 7. I try to put on hair again a bit, just so it's attaching a bit better. And we also have less movement of the last electrode before we come to the ground. Dreamy agrees on the measurement. And now I'm going around. I'm placing it on the position behind the ear. 1, where we reach here. There, higher, because we could reach the muscle with that, which is so good, because then with the muscle we would have a lot of muscle here on the ground. Behind the ear as good as possible. Good. Okay. Now we're actually good to go. I will also again stick here the cables on the side. So, and now we have all electrodes attached on the right row. Outside. And with that, I can actually start software. So sorry. Here. So first of all, I will put the amplifier on top. So maybe Tara, you can also film that. Here. So this is the amplifier. I just need to plug it on top, very simple, with the magnets from the, and then it turns on if you see the light. Maybe it's a bit hard to see here now, and now you can see it. It's flashing up and down. And with that, we have the contact. So we have the amplifier attached, and we are ready to start. So okay, let's see. So now I will actually start. If Sarah can maybe film again a bit the amplifier. I will start up the software and then share. I got data. So you could see actually the setup was quite fast. So we didn't have to do a lot of preparation. So I was actually already um, seeing some labs that did force measurements, and they did actually a lot of preparation, including shaving the skin, which obviously is not the, I mean, obviously also is again taking time, and is not the easiest for the horse itself. So again, from here, which is not the easiest if you need to shave the skin before or the hair away. So in our case, we need to do that, which is really nice. We can attach the electrode gel to the scalp. That's everything that you need to do. Amplifier, and then I will stop the video actually and share on my screen, because with that you can see the EEG on scope, which is quite important. So okay, I have here the 4-channel connected actually. So first of all, I will show you, I'm using here now the 4-channel amplifier. I will share just the screen, you can see. You can see here now the data from a 4-channel BCI core, because I attached here the 4-channel BCI core. And I do see it already. Can you see my screen?
We can see it.
Okay. So I attached the 4-channel BCI core. You can see the EG now first 4-channel. You can also see the movement here. I also have an 8-channel BCI core that I will attach afterwards to show you how this works. So we have here the EEG from channels, and when there is a lot of movement, you can see also higher frequencies. We filter here with a 1 to 30 hertz band pass and a cascading notch of 50 hertz. And with that, we have actually a quite good EEG here. Channel 1 and 2, maybe I need to check the attachment again a bit. And yeah, we have a scaling of plus minus 50, so it looks like that. So we also maybe have a channel 1 eye blink, which
Can
is,
you ask Trimi to blink?
yeah, maybe we can close the eyes. I mean, actually, the blinking is a bit hard, but we can try to,
Or
because
maybe
I'm
you can
trying.
give him something to eat so that we see the
Yeah,
EMG
and you can
contamination.
see,
But first get the EEG data clean.
let's try a bit
So
of
channel 1, 3, and 4 are already pretty good. The second one is floating, as you can see.
yeah, that's nice.
So,
Dream is
so
interested in the treats already, so I'll give him some, because I already took some. I have here channel 2. Yeah, this looks like it.
that's it. Now you can see that Leo lifted off the electrode, so the signal gets lost as it should be. It's just floating and then it's just picking up nonsense. So non-attached EEG channels are completely meaningless.
Now
Now
I
you can
touched
see it
again, it
looks
looks
already
good.
better.
Yeah.
Oh, it's
I'm
nice.
recording here from the 4 channels, and yeah, in this case, because I used the BCI for 4, in that case we have the lower 4, so you can actually see eye blinks a lot, particularly in channel 1, because this one I placed a bit closer to the eye actually, and also Dream is very interested in the EEG now. And
Let's wait for another eye blink so that we see that the video is on the eye. That's good. Now he
now,
blinked. You see,
yeah,
that's the EEG artifact.
exactly. I can also mention it if you want. Now he moves a lot, so we have some movement, but maybe with the eye now was a blink, especially in channel 1, because this one is just close to the eye. Yeah. So this is what we see in this case.
Susie, can you maybe feed him something so that we see the chewing artifact?
I have a really good crunchy thing. So now he starts to eat.
Is this what you were eating before yourself?
No, I did not, but it looks very delicious
I
and
think you confused the dog food with the human food.
yeah, this so uh, so Dream is actually quite easy and yeah, like the measurement. So what I'm doing now, I'm actually selecting BCI Core-8 now, and I
Leo,
will
maybe first we can also, if maybe it's possible to see some alpha, can you somehow shade his eyes? Maybe Susie can do it. So if
let's
the
try.
eyes don't get visual input, we should see some alpha waves.
Do you think can do this? Let's see how Dream is going.
Yeah, here, alpha is coming. Now remove it, yeah, we have higher amplitudes, and when you remove it, you can see it gets smaller again. Nice.
Actually, I
That's
want to record this data because that's quite.
that's
I
horse
will.
alpha.
So I will record. That's also how we generally record here with the recording button. And now I also have the data saved, and I want to try this again. Let's see. Easy again.
Yeah, it's
Not so
coming
long,
again.
but he
But you can see the rising amplitude. The EEG is about double size. And interesting is also the magnitude of the EEG. So this is roughly plus minus 60 to 80 microvolts. And can be explained by the distance from the grounding electrode to the other electrodes. So maybe, Leo, you can show again where the ground is located.
exactly. So the ground is actually behind you here on the left side.
So the distance is like 20 centimeters.
Yeah, it's quite
Mm.
it's quite. So
Okay,
you can also see how Dream is interested in the leg and
in the food, not in the neuroscience.
no, I mean I don't know. He's watching also the screen a bit, but he's more. Okay, so let me maybe Sarah can now film again a bit in detail how the setup looks, and I will stop share and connect here the 8-channel BCI and share again. Stream is interested, but it doesn't not in me. So let's go and take off the amplifier. You can also see how easy this is. So I just take it off, these magnets attached, and then I'm taking show it first to Dreamy, and then attaching the next one. It's blinking in blue if you could see it on the video, maybe not. I try Arabic Alex. And
So how old is Dreamy? Because he's getting white hair.
how old is Dreamy, Susie? 8 year, 18 or 8? 18. Yeah, 18 years. Not
Pretty old for a horse. Yeah.
young.
Leo, now we lost you.
You lost me.
I lost the screen, so the video
Ah,
and
okay.
your screen sharing.
Okay, let me do that. Cannot start video. Let me try that again. Now I'm back, no?
Yeah, the
Okay.
video is back. So Dreamy and Susel are back.
Okay. So, but you can hear me, no? Then I will share my screen now again. First, connect.
So Dreamy was mowing our garden in the afternoon, which is very nice. And he can do a lot in the afternoon. It's amazing.
Yeah, he was actually eating up the grass. So yeah, you can see here now the 8 channels. We have channel one. I think it must be best connected. I'll check it in a second. But you can see the other channels are pretty good. So maybe we can try. I will record now. So maybe we can try closing the. Oh, you maybe you could hear or see it moving now. Um, the feet. So you can clearly see that with some higher EMG. Now I stopped and Maybe you can try to close the eye if you want to.
It's
Or
beautiful,
should I?
amazing.
I can try.
Mm.
So we have now closed eyes. So we try that. Maybe you can see them higher amplitude. Oh no, again. And what I also want to show you is actually the paradigm presenter. So how we perform the experiments. So I will just show you shortly now how we can load an auditory paradigm. I'm not sure if you can hear it then also, but I'll just try a paradigm, standard output paradigm that we until last time. So screen.
You're doing the blondie check with Streamy?
No, we do now just an auditory experiment. Maybe you can also hear it a bit. And now you can also see the triggers coming. So the blue lines are the triggers. So you can see them here. And this is what we need afterwards to analyze the data for the EPs that you have seen before. So look, Creamy already recognizes the sound because we did that experiment quite often. And she is excited because she wants to have some treats after the experiment. For now, she needs to leave a bit at least. She still lives, but I will give her a treat, even though it induces some. Thank you, treat.
And Leo, for the EEG data that you are saving, it's also filtered between 1 and 30, or you are saving the whole spectrum?
So we can do both. So what we actually mostly do is always save the raw data, and additionally, we can also save the processed data. Typically, we save it as a CSV or it's a PDF file, so we have all the options. I already started the saving button, so here the blue circle button. Stream is not ready. Maybe you could even see the EPs.
Can you maybe put the microphone a little bit closer to the soundboard so that we can hear it?
I can hide, turn, and hear some beep.
No, beforehand we could hear it, but now we don't hear it.
Yeah, unfortunately, maybe the sound.
Okay, not so bad. We see the triggers.
Yeah, just like standard and deviant tools. So we have like a low speed 5 times in the ratio and a high speed 1 time. Our oddball, and just takes a few minutes, and we can run this a couple of times. That how we all.
So how comes that the Trimis EEG looks like your EEG?
So yeah, basically we were quite similar. No, I would definitely say, I mean, when it comes to analysis, we have very similar behavior also of the auditory evoked potentials, also talks, and we have just a bit more higher amplitude components, which are probably for movement, especially with ears, because we always place the electrode somewhere behind the ear, close to the ear, and in dogs, there's a lot of movement. Um, of course, there is also the option to just place the electrode centrally, for example, but we also don't want too much EOG inside in the reference, so we also don't want to place here.
So now we are even getting alpha bursts. Trimi gets relaxed. So here you can see, for example, in the middle of the screen, some alpha bursts.
Yeah, he's very happy.
Hmm.
Now you heard a loud noise
Here
in the
again.
background.
Yeah, somebody's asking if Trimi sleeps standing or he lies down. Do you know?
I guess he's standing most of the time. Susie is really sleeping standing or lying down or both. So he does both. He can do both. But I'm actually, yeah, this is actually quite interesting because also a big research interest in making in doing EEG is about their sleep hygiene. So this is very important.
So
And,
can Suzel now jump onto Trimi and ride around a little bit?
no, I don't think so, but I guess she can walk around a bit and we can check how this behaves. So I would suggest this right now. So
She also has another horse, which is a jumping horse. So next year we are doing the parkour.
we can
Mm,
walk around it
mm.
and see, like the wireless device, how it behaves. So I'm not sure how far we can go, but you can probably see now the movement architect. Sarah is filming it. Yeah, so we are actually outside, so that's not the best case for a Bluetooth Low Energy because we don't have a lot of reflection on walls, so we prefer it inside. But still, you can see you can do all the measurement outside, freely moving, and this is very interesting because you don't need to limit here their normal behavior or sedate animals. And yeah, that's the signal. We see also the movements and biting.
So why
Christopher,
is there a blanket on Trimi?
why is there a blanket on green? Because of the fly. And Christoph also asked if you could do a little ride with her in the car, but you don't have to. No, so I guess we should
Here
not
was
do that.
nice alpha. Did you see it at second 426? Beautiful alpha burst.
I can try again.
Mm.
Let's try it.
No, it just occurs on when he's relaxed.
Yeah, I mean, I would definitely suggest that there was high amplitude when I closed the eyes, but he's now.
But interestingly, we don't see the chewing. So when he's eating, we don't see the muscle
Uh,
contamination.
because we have the I placed the ground then particularly not on any muscles here, so behind the ear, so you most probably see more ear movement. So when he's
Mm-hmm.
listening to different, like I'm doing now, then he's like moving the ear a lot, so you probably see that more now.
Mm-hmm. Yeah, the ear muscles are closer. And for
Yeah.
humans, of course, the muscles for the mouth are closer to the EEG electrodes, and especially the ground where we are recording. And for the horse, of course, the muscles are much further away from the EEG electrodes. That's the reason why we don't get the EMG in the horse, which is, of course, a big advantage.
Mm. Yeah, and so I think we showed most of the setup and how we would handle it, and it's actually quite fast and actually time. Um, we can already start answering questions, Christopher. We can also do some more movement experiments.
One of the questions is how do you choose where to place the EEG electrodes? They are more on the left and right side. So what can you do in terms of channel selection or position selection?
So of course we try to adjust to like research, so movement, so some listed. So of course we had tried to go with maybe camera in the question. We try to go with standard positions. So we look for in literature where the positioning should be. We also build our caps accordingly, and we want to be comparable to literature. And we of course try to be, as close here to the horse brain. So as I placed it today, actually was a bit too lateral, I would say, because in this setup now, normally I would place it more centrally, both rows. I also have to say the right row was a bit more lateral that I'm used to place there.
Do you still have the EEG on?
No, I already switched off the device right now.
Ah, too bad, because we wanted to do a real-time experiment. For human beings, if your mother shouts your name, is actually the best case for a strong P300 response. So we wanted to test when we call out Trimi's name, if we can see the evoke potential in the EEG. Fixed and sealed the EEG electrodes on the skin of the dolphin. So it was waterproven. which was very interesting. And then they had auditory stimuli underwater. And this is, of course, super interesting for dolphins because they have a very good sonar system. And researchers are interested in how the auditory cortex is working in these guys. So that's a very nice use case. If you consider dogs, they have, of course, a very good nose. So in this case, it would be super interesting to figure out how the smelling system is working in these animals and to learn more. And of course, with the BCI interaction, you could maybe even utilize the dog's nose for figuring out a little bit more. You could classify the data and just achieve a little bit more. Any other species that you have seen? You went to Krüger National Park. You put it
Yeah,
on rangers, right?
I would put it on us humans, unfortunately, not on the elephants or the rhinos or all the other impressive animals there. But we have a lot of interest and requests for particular animals. So, yeah, seals, for example, when it comes to animals in the water. So, making enclosures that are waterproof also for the electrodes is a very interesting topic. As with the dolphins, because you said they had to go to this plate to just be fixed and stay there. But with the wireless system, you could maybe even give them some more freedom there, and also a lot of different animals and species.
Yeah, in salt water, you are actually getting two problems. First of all, it's conductive, so it's shortcutting the electrodes, and then you're not measuring anything. And secondly, we use 2.4 gigahertz, so it doesn't go through water. So there's no chance to use it underwater, except if you fix everything onto the dolphin or onto the fish or onto the diver, and then they can dive with the headset, and it would transmit inside the diving suit or helmet, whatever you design.
This would be an idea, or a little boat that is floating on top of the water with some cables that go down the dock.
So thank you very much for the nice session. Maybe you should Remy again if he's still here so that everybody can say goodbye.
Thank you to Primi. Thank you, Susanna.
Thank you for coming.